Okay, welcome everybody. We resume with the last afternoon session. The session is about open science and reproducible research. It's going to carry us with 5 speakers until the coffee break, and after the coffee break we return again for a panel on reproducibility. I'm very excited about this session as well as about the panel. I hope you are too. And we start off with Carole Goble, who's going to speak on about Seek for Science, a data management platform which supports open and reproducible science.
Thank you. Thank you. Okay, so I've got the graveyard stuff after our exquisite lunch. So I'm going to talk about this Seek for Science platform, which is a collaboration between 3 major organisations, University of Manchester, the Heidelberg Institute of Theoretical Science, and the University of Stellenbosch. It's an European-based project, European-funded project, and you can ask me afterwards how the University of Stellenbosch in South Africa is part of Europe. It's one of those weird funding arrangements you can get into. So what we're interested in is systems biology. So systems biology 101 is a mixture of experimental data collection, hypothesis-driven and data-driven research, along with models, building models, linking them to data, linking them to the experimental practices, model construction, model analysis, model validation, and model prediction at the end of the loop. So this is the systems biology kind of classical loop that we undertake in the research. So the sponsors of our data management platform are 2 major projects in Europe. One called Systems Biology for Microorganisms, which is a pan-European program funding program, which 16 multi-institutional consortia took part in to study various different kinds of microorganisms in a systems biology way. And it was run as a series of independent projects in 2 rounds of funding, and it encompassed a great deal of organizations. And the second one is what's called a BMBF Gross Project, which is a German program of 45 organizations divided into 70 different task force groups who are looking at the virtual liver. And they're particularly looking at virtual representations of the liver from a point of view of different scales, and they do a lot of imaging. So what the funders wanted was to preserve results beyond the end of these projects, to organise and link the data and models and processes together, hence the reproducibility aspect. So you could link the data to the models and the models to the processes and the models to the data, and you could link the data that was used for construction of the model, the data that was used for the validation of the model, the data that was used or was predicted from the model, and this would all be different. And also to collect, for example, the standard operating procedures associated with the experimental data collection. They also want to promote the notion of exchange between the different projects funded in these initiatives, hence the open bit, and to share and disseminate the results beyond the end of the projects and improve also the quality of data curation, model curation, and standardized practices, and basically to pool capacities. And we also had to deal with all the home-brewed systems that different projects had. So I'm going to see Monty Python at the end of next week, by the way, live. Just thought I'd brag about that. I've got really good tickets at the front, yes. People. So what do we have to deal with to do this? Well, the first thing we had to do was those really difficult bits of any kind of data sharing platform, which is the people that use it. This is the hardest bit to crack. We're talking about dynamic distributed groups of modelers and experimentalists who are mixed teams. They have their cherished homegrown and unstable data management solutions based on wikis and CMSs and databases that they've forgotten how they work, and spreadsheets and file systems and so on. And they also have very strong ideas about who can see what when. Amongst their own consortia, amongst their own organisations, but particularly to the public. So the funders on the one hand wanted open, and the researchers on the other hand wanted control. We also have very many different kinds of content to deal with, the locally, privately hosted private repositories that people had, public archives that they wanted access to, for example, Express, Geo, all these usual ones, Sabio Archive for reactome and enzyme data and so on. They also needed to represent samples and specimens. Standard operating procedures were very important because they needed libraries of these to understand experimental data collected. And we were dealing with small data, effectively in spreadsheets, and big data like mass spec data in specialist repositories, as well as models of different kinds. A mixture of SBML, so that's the standard markup language for many of the ODE models, but also just great lumps of MATLAB. And we wanted to also manage models and data while it's being in progress, scientific progress, so it was being versioned and tracked and so on, and also to deal with it when it was being published, so it could be cited in each publications. So we started in 2008, so it's been going a long time. We built something called the Seek platform, which is a Ruby on Rails, which is currently in 3.2, BSD, openly licensed platform. And it's basically a web-based app a web-based application to handle the relationship of all of these different things. So it has yellow pages for people, experimental details broken down into a hierarchy I'll briefly show you about, and various different assets. So the projects could contribute and share their resources and also retain their data and retain their models and link them in this platform. A core part of the Seek is its cataloging capability. So this was absolutely fundamental because different projects and different teams had different data sets, and what they wanted to do was catalog them. We wanted a shared catalog, but not necessarily to move the data out of the research groups or to move the data out of public archives. So the cataloging is the heart of the Seek system. So I want to track specimens, I want to browse experimental data without downloading them. I want to see how data and models and standard operating procedures fit together. So this was absolutely critical. So the Seek is what's called really an aggregated asset infrastructure. That means the majority of what it holds, it doesn't hold. It doesn't keep. It's actually an Uber catalogue that tries to span multiple other catalogues and was designed in that direction. So it uses the ISA structure, and I'll briefly mention that in a minute, which is Investigation Studies Assay structure, which has been developed out of Susanna Sansone's laboratory in Oxford. And it uses that as a framework to be able to hang off data, hang off models, hang off publications, hang off samples, hang off links to external databases, and to bridge and interlink the different resources used for a particular investigation or a study or an assay. So this is important because it means that we can incorporate and reuse other people's infrastructures, other people's data sets. So our reproducibility card kind of looks like this, that we have this catalogue that bridges different kinds of resources, is very standards orientated, and I won't go into the details of these. I'll just briefly walk you through a few of the things that are interesting. But you can look at this when you go down through the slides. As an architect, sort of architecture, it's a confusogram. It's a picture of the idea of that what this gets across is, first of all, it has an API, of course, so you can use it as a normal resource. It has some local repositories for data and models, so locally versioned models, for example. But the important things are the ends, that it's kind of bridging out to standard tools and to the community's local resources through an extract an ETL platform, actually, an extract, transform, load platform. Because of that, it plays nice at doing gateways to integrate into other tools. We have tight and loose coupling, really leveraging the Rails plugin framework, and we have a metadata framework in order to be able to register things into the catalog, because that's the core thing. And all of these tools are incorporated, all these resources are incorporated in the standard Seek, which is implemented for the SISMO project, and we're about to incorporate the OpenBiz project, which is a big platform developed by Systems X in Switzerland for doing large-scale data management, particularly taking data directly off instruments and managing long pipelines and compute-intensive pipelines. So I won't have much time, so there's lots of data. You can have different kinds of data, different public data set samples, as I said before, all tagged with controlled vocabularies. It has models in it, and this is where the reproducibility aspect comes. So we have support in the system for being able to do standard markup of the different models in the standard different schemas for the models, SBML and ZML in particular, for the linking of data to models. And we have onboard tools to compare construction and validation data with the models, and to run models with parameters values taken from spreadsheets and then compare the results. So the important thing here is the packaging of data, models, and collection procedures together. So that's what I more or less just said there. So we use the framework, the ISA framework, as a way of organizing all of this. And I've already said this as well. To give you a kind of little look at this, here's a experiment, here's a model that you can, with some description associated with it. This is the kind of, you can see the ISO exposed in this. We have a folder view and a network view, and I've done the network view of the actual investigation, the studies, and the assays. And then you can drill down to the data files that are associated with that assay and look at the relationship of the data files to models and the data files clustered together, and the relationship as well to publications. So I'll let you have a look at that later on when you look at the tool. So the metadata framework that we use, because the cataloguing is absolutely critical, is based on something called the just enough results model, which attempts to take all of the well-known MIBI style minimum data models that have been widely used for microarray proteomics and enzymic reactions, for example, and turn them into just enough. So on behalf of our scientists, we stripped out a lot of the maximal stuff in the minimum information models and reduced it to a description that was sufficient for each of the different data types, but also brought out things that were sort of the relationships between things produced in experiments at a higher level, what type of data, what was measured, the values in the data sets, and treatments applied. And we have that as an ontology, which is included in BioPortal. We set up for each of the data, omics data collections that go into the system. We have helped our users set up spreadsheets for collecting the data. This is for particularly the smaller data, using the right field tool, which basically each spreadsheet template represents a different kind of omic data, and then we've instrumented these spreadsheets with controlled vocabularies, so when they get distributed, the data just goes, and as you enter the data, things that are drop-down menus turn out to be ontological controlled vocabulary terms from the controlled vocabularies of the community, and so we're collecting very rich data that way from our from the things that are spreadsheet-based, as opposed to things that we can directly collect off instruments. So what happens is that we have these standard templates, and there are some non-standard ones that the scientists use themselves, which are actually cunningly structured. So this is annotation by stealth, where the data is collected in this rich way because we produce the templates and we produce the control vocabularies behind them. These are ripped apart by when you upload to the Seek, or when the Seek goes and grabs them from your platform, and then combined with the models that have also been annotated in the appropriate control vocabularies, and then we can mush together, we can auto-catalog given this metadata. And we can also spit it out, combine it, but we can also spit it out in the combined open modeling exchange format archive. And I stuck in the research object slide. So I'm on track to get money if I was an American, but I'm not, so there we are. But I partner with somebody who's American. We've been working on research objects for a long time at Manchester, and the combined archive, which links together models and data into one kind of structure, is an example of a research object, and we've been spending some time mapping the combined archive structure to a standard, a model of research objects that we've been promoting out of other projects, which uses standard mechanisms for doing aggregation, provenance, identity mapping, and so on, all from the library community. And you can upload these into Zenodo or GitHub and get DOIs for them. So to finish off, this is an open reproducible research group session. So the whole of the SEEK is about being able to upload and gather together and retain results from these different projects to make them open, but particularly this idea of grouping together and packaging models, data, procedures, and so on as a unit, and then be able to explore it and export it and identify it as a unit, rather than just a separate kind of, here's a data file, here's a data file there. But the openness is an issue. You can probably see here something called hidden files, or maybe you can't because it's hidden. It's a hidden file grayed out, and that's because we have very fine grain management for the owners of this data and owner of these models about who can see what when, and it's a challenge to get people to expose data, even if they're published. So we're still working on that. That's still an issue, and Z Titus and Phil's talks for why that's an issue. So within these projects, we've had to really develop sharing policies, fine-grained permissions, protocols for management of and transfer of ownership, and all these kinds of things that understand how research groups work and the life cycles of data within those research groups. So to finish up, I'll just mention this, that this is a very open program. It's actually, this software is all the underpinning the BioVel project that Rob mentioned earlier, so the software is actually highly configurable. So we've just been funded again, yay, to build FAIRDom, which is an open facility for European systems biology, and this is to open out. These different installations of SEEK have been used by many different other projects, and this is to build a EuroSeek. So to make this a European place for systems biology data and models, the platform is going to be integrated with the OpenBiz platform from Switzerland, and we're providing a complete facility for that, and including community building, including model carpentry. So we're funded to establish model carpentry to go along with software carpentry, and we have a systems biology developers foundry workshop in October that you're all welcome to go to. So thank you. Now I'm being dragged off by Hilmar. Thank you.
We have time for maybe one question.
I'll just pass it off in the back. I was just curious. You heard all the talks this morning about building commons. You guys really have a commons.
Yes.
And I feel like it's turning into standards, right? Like everyone has their own standard, and if we just have 10 of them, and maybe they'll come together eventually. What do you think the future is? You can't hear me.
So what do I think the future of commons are?
Is bringing the commons together into one technical place where we can have a real commons for everybody.
Yeah, this is actually, I realized during the talk that this is during Phil's talk that this is a commons, yeah, that was funded by the research councils thought of this and then asked us to build it, despite the scientists not wanting it, and but now they do, hooray! And they did it in 2008, so they were well ahead of the curve. This is all built on just normal standards. There's nothing we've invented here. The idea here was to be completely standards-based, from the systems biology standards to the omics standards and so on. And we can export into other commons. So the point is we're able to import and export. That was the point. So we could team up with the US commons, for example, and we should be able to do that pretty straightforwardly. We have tried very hard to do nothing clever.